This is episode 116 of the Immunology Podcast, Allelic variation with Dr. Robin Orozco. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Robin Orozco from the University of Kansas on the podcast to talk about her research on the role of allelic variation in immunity. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
STEMCELL Technologies would like to introduce you to human immunology news. It covers everything from immunotherapy, autoimmunity, and adaptive and innate immunity. Human immunotherapy news keeps readers current with the latest news, research, policy, events, and jobs relevant to the immunology community. Subscribe for free at www.humanimmunologynews.com.
all right. Well, how
Hey,
are you, friend?
Jason. What's up? Happy New Year.
Oh, thank you. Yes, it is that time of the year this year. It moves every year because it's a lunar calendar.
It does?
Oh, yeah, it's a lunar calendar. So it moves based, compared to the regular calendar all the time.
Oh, that's very interesting. And how many years are you guys on already? Like 6,000?
Let me look.
You don't know?
No,
Are you supposed to know that?
5,786.
Good. All right. Well,
Yeah.
very important. Well, I hope for another 5,700 years, whatever.
Yeah, that'd be good.
That
Keep
would
it
be
going.
nice. But they're lunar years, right? Are they shorter? So they're shorter
No,
years?
it's still a full year. There's 12 or 13 months because it's a lunar calendar. And so to
Oh,
leap year every so often, they do a leap year.
all right. Okay.
It's similar, very, very similar to the Chinese calendar, actually.
Ah. But the Chinese calendar also changes? I thought it was always like in January.
It's sometime in January, but it's not always January 19th or whatever, right? It moves. Just like Easter moves, because Easter follows the Jewish calendar,
True.
right?
True.
So it's,
Okay.
it's a lunar thing.
We should all do lunar calendars. I think it makes more sense.
Yeah.
Like the revolutionary calendar, like the French 13-month calendar.
Yeah, you could just do 13 even months. That'd be nice too.
Yeah. They should have done that. They should have kept that. We kept the meter, but we could have also kept the, revolutionary calendar. That would have been kind of nice. Anyways, talking about revolutionary, any revolutionary research you want to share with me today?
I don't know if it's revolutionary, but I'm going to start with Staph aureus.
Staph aureus, our favorite, non-favorite bacteria. Sure. Hit me.
Yeah, yeah, there's no good segue about I want some staph infection. No, you don't. All right, this is Staph aureus exposure during cutaneous antigen sensitization causes basophil and interleukin-4 dependent exaggerated food anaphylaxis. So first author is Mrinmoy Das. Last author is Raf Gaya. It is in Immunity on September 25th. I will say this is an interesting paper. It hits hard early and then kind of meanders, and I get confused about some of the other stuff they look at. I think they're hoping for some greater connection to all things that didn't pan out. But it starts cool. And so let's be positive here and start with the cool. So we know food allergy sucks, right? And people get it and it's unfun. And there's a known thing that having Staph aureus on your skin, especially ones that produce super antigen, so enterotoxin B, it has higher concentrations of Ig antibodies to food antigens. And colonization and food allergy are associated with each other, independent of disease's severity. So having this thing on your skin leads to food allergy. What? That's weird. We also know, by the way, that oral allergies can often come from getting sensitized on your skin. So that's another fun thing. My wife has that from zucchini, actually. She used to garden, and then within my garden, it was my fault apparently, and now has a zucchini allergy to eating it.
We did discuss that also in a roundup, right?
Yeah,
we had a paper about that.
Yeah. So it's like everything ties back together, especially to my wife. At least for me. So that being said, they find to start with, so they take patients and look at what's going on. And yes, it correlates, but they find IL-4 concentrations are elevated in patients with oral allergy, right? And so that was the first thing, and ectopic dermatitis. So these go together. So people with atopic dermatitis have more food allergies when they have more IL-4 and the staph aureus, but not IL-13. So they linked it to a cytokine, which is the first part. Like, okay, well, how is this actually working? So they found that it results in exaggerated. So they went to a mouse model, which is an ova model, which using tape stripped skin. So you tape strip a skin and then put ova on it, and that's skin sensitization. And then they find that causes anaphylaxis to oral ova. Okay, so that's the model for the rest of this paper, because we're not going to do this in people, right? Because that's not cool. So they find in the mice elevated IL-4 when you have the staph colonization on there and use the system as the next step. Okay, so we have something similar in the mice. They find that it also, even when they do the ova thing, you can do other antigens, and that and then they use a different antigen, which is this thing called TNP-BSA. It's a trinitrophenyl antibody, and then something tight, and then the trinitrophenyl BSA. And so if you'd use that as a second system, the mice also, even though they were sensitized to ova with staph, they're also sensitized to that as an oral antigen. Right? So if you do a second insult without the sensitization, with sensitization is something else, it also makes it worse. So this whole system is a general priming, not just to the antigen du jour, in this case the ova, but in general. It doesn't exaggerate using the same thing with this TNP-BSA, but doing it IP or ID, intradermal or interperitoneal challenges, doesn't cause increased anaphylaxis. So it's only an oral anaphylaxis effect, which is interesting, right? So it's an oral mechanism. And then they of course, then try to figure out what's going on. So they take splenocytes and they look at cytokines and eventually, I think, RNA-seq, and find it's a systemic antigen-specific systemic Th2 response. And that drives increased test in the IL-4, drives increase in intestinal permeability, which probably leads to the sensitization, right? If your gut's more permeable, that's a signal. And it's worse with the bacteria, right? So it generally happens, but with this extra IL-4 brought on by the staph aureus and the supersensitization there, you have more of it. And then they looked at where are the immune cells going, and they find DCs from lymph nodes of skin, draining lymph nodes, are the ones that promote the Th2 polarization and demonstrate increased IL-4R signaling. And then they have this left turn at basophils. They're like, well, what else is happening? And then they find that basophils accumulate in the draining lymph nodes, and that potentiates the polarization as well. And the IL-4 from the basophils and it's IL-4 from the basophils that do it. And that the basophil of influx to the skin from exposure to SEB is dependent on CD4 expression by keratinocytes and keratinocyte-derived IL-33. So when you put this all together, it looks like when you slap this antigen on the skin, you have more basophils going through an IL-33 dependent mechanism in the surrounding tissue for the super antigen. When you then have a skin sensitization to an oral antigen at the same time with injury, especially with this bacteria present as a colonization, you have a hyperactive IL-4 response that leads to gut permeability and oral allergy. And then this IL-33, and then there's a feedback loop with IL-33 driven T cells that drives the basophil influx by IL-3. So it's a whole IL-33 in the keratinocytes to IL-3 in the T cells to pull basophils in, which boosts the Th2 response with IL-4. So they try to map it out all, but I think the point is they're really starting to understand the mechanism of how this known superantigen, which is associated with atopic dermatitis and oral allergy, works. And it's through this IL-4 dependent mechanism that is ultra primed when this bacteria is present. It leads to a leaky gut. Leaky gut equals more allergy, but only oral allergy. And that's why it's only oral, because it has that gut leak effect.
So it really is two independent sensitizations. It's just that the one in the skin makes the other one more likely.
Yep, exactly. That super antigen makes the response tuned up, and that tuned up response leads to more anaphylaxis.
Yeah. Yeah. Okay. All right. It's very interesting because yeah, to understand where these allergies are coming from. Um is there some kind of clear or suggested therapeutic application of this knowledge to prevent? I guess once it's done, it's
They know that
done.
you can like wash people with bleach to get rid of the staph, but they don't actually land on like anything that's reasonable. Like, there's always the discussion section which talks about, well, therapeutically, you could do this mad science thing, but it's really about the mechanism that's known effect is occurring,
Mm, yeah.
right?
Yeah. Well, I guess that's yeah. It's another example of like everything kind of impacts on everything else. It's just you cannot escape the complexity. Oh man, we're screwed. All right. Uh I will follow up with a paper full of pretty pictures. What
Oh
are some of my
boy.
favorite type of papers? And also has some cute videos. Oh, all right. So my first paper for today is was published in Science Immunology. First author Yukako Asano from corresponding authors Gillian Griffiths, and they are at the University of Cambridge. Um, and the paper is called Nuclear Polarization to the Immune Synapse Facilitates an Early Transcriptional Burst. And basically, what they show using some very, fancy, very close up microscopy and, confocal microscopy, some in vivo, some a lot of very clear microscopy, also electron microscopy. They describe an interesting phenomena that happens right after the formation of an immune synapse, right after recognition of an antigen-specific T cell of their target antigen. So here, all of this, I think, is based on an OT1 with OVA expressing, OT1 cell with an OVA expressing target cell. And they use 3D live imaging to follow the very early times in this synapses and this immune recognition, and they find something that was remarkable, which is that very early on, I think this is. I'll go into into the actual times in a second, but very early on, one of the very early phenomena that they see is that the entire nucleus of the T cell seems to kind of gravitate towards the immune synapse. And this is not necessarily something expected. And they show that it's kind of a pretty well established thing. All happens really within the first few minutes of this interaction. And they see it over and over, and they do a lot of microscopy showing that this is really something that happens even before other well-known events downstream of the immune synapses. This happens. Um, so we already know that one of the things that happens, but already around 5-6 minutes after the start of the immune synapses, which is a uh, transportation of the centrosome, which is this little organelle that is important for the organization of the microtubules, and it's part. It moves around at certain times, particularly during mitosis or cell division. But also, as it's already been shown that um, around 5 minutes in, you have this translocation of the centrosome, which is usually in kind of one of the sites of the immune cell, right next to the immune synapses. But even before that, around 3 minutes, not even 3 minutes in, you have um, this recruitment of the whole nucleus really close to the immune synapse. And this is something that they observe, and they try to understand what is the implications of this translocation. And they show that it's um, important for the expression, the very early expression of certain effector molecules, particularly TNF and interferon gamma. And so, what basically that they hypothesize is that this translocation of the nucleus allows for a very quick transcription of these elements, interferon gamma, TNF, but also CD3, which is increased molecules. CD3 molecules are being produced during the immune synapse, and that this is important for the function of the T cells because they show by disrupting this nuclear polarization, which they show depends on specific myosin 2A, which is a specific mechanism for kind of moving. This is not depends does not depend on microtubules, but on myosin. And if they prevent the myosin from moving the nucleus, they can impair production of certain immune cytokines and ultimately reduce the effectiveness of the immune synapses. So it really seems like this translocation of the nucleus means something. It's important for the correct activation of the T cell and the effector function. So yeah, I thought it was really cool. They have a lot of very they detail microscopy. They show, there's like this Golgi ER formation right next to the nucleus and the synapses. And that this is not just something that happens, but it's actually important for the whole um immune activation. This happens after the influx of calcium. There's a direct consequence of a TCR activation and the clustering of the TCR. So right after, this is what happens when the nucleus gets closer to the immune synapses.
That's super cool. I was going to ask you if they made sure that the function was there, and they did. So good job. Good job for viewers.
Yeah, it was pretty cool. I have to say, now that I'm discussing this, I don't completely understand. So how so they mentioned
myosin works
that
on actin. Myosin actin networks.
Yes.
And so block that. And so if they block, and that's calcium based. And so that's how they, you can move things based on microtubules or on myosin. Myosin's how muscles work.
Right, exactly. So it is not microtubules, because they show that if you have compounds that prevent polymerization of the microtubules, this still happens.
Right.
So you don't have other changes like the centrosomes, for example, they don't cannot migrate, but the nucleus does.
Yeah. Well, this makes sense.
Yeah.
Myosin can do that.
Yeah.
All right. Well, I have no segue because mine's about CAR T cell therapy. I'm stealing your shtick, Brenda.
Yeah. Stay on your lane, Jason. What's wrong?
I don't know.
Not enough, microbiome papers for you?
Not this week.
Yeah, sure.
All right. So we got in Nature coming out 24th of September. Systemic discovery of CRISPR-boosted CAR T-cell therapies. Systematic, rather, discovery of CRISPR-boosted CAR T-cell therapies. First author and co-corresponding author is Paul Datlinger. Last author is Christoph Bach. Okay. So they came up with their system called CellFi, a CRISPR screening platform for enhancing CAR T-cell therapies across multiple clinical objectives. I think, and I don't do a lot of CRISPR screening, so I kind of have to just believe the authors here, is what's cool about this is that the CRISPR screening works really well. And so they spent a whole bunch of time that I probably don't appreciate very well working on getting the CRISPR screen to work on top of a regular CAR T cell therapy. So they used something that was very close to Kymriah as their model system, and then Jurkat cells as a control of like, look how much we can do in your cat cells. If we can get CRISPR screening to match your cat cells or almost, this CRISPR screen really works, which they did, right? And so they developed this CRISPR screen by some what looks like clever ways of introducing the guide RNA by mRNA. So they have the mRNA make the guide RNA. And to develop CRISPR editors, and they also used mRNA to develop CRISPR editors. So long and short, they try to make sure knockout worked. They controlled it with an IL-2A receptor knockdown. Long and short, they get this sucker to work. And then they first validate it by saying, if we do a total genome thing, they do a total genome thing, right? And do we see what should break? If we knock out a gene that's important for proliferation, does that happen? Does proliferation go away? And guess what they do, right? And if they knock out genes important for stimulation, then that goes away. So it captures intrinsic, and also what is cool is they capture intrinsic limits of CAR T cells. So we know CAR T cells show slower expansion upon CAR stimulation than TCR stimulation. We know that it'll acquire CD19 antigen and then go through trogocytosis. We know that CAR T cells upregulate certain markers of exhaustion. And if they find out when you knock down any of these, the cells get better, right? So like, okay, cool. Things that we expect to work, that we should expect to have gain of, expansion with knockdown, they do. So I actually like this. They're being logical on their validation. And then they do a genome-wide screen of binding across the whole thing, and they find things that are very typical. But then they did this in mice with xenografts as well and showed that it also works and that they can get the same signals and boost signal there. And then they go back and say, okay, we're going to look at the cells system. And then we're also going to look at the mouse system and see what really has an impact on tumors that's new. Right? And so this is where they get to some new things. So this is FAS as a cell death receptor, so not too surprising. Its expression has been shown to limit CAR T cell efficacy, but they find that. PRDM1 is an epigenetic regulator, and it's known to enhance CAR T. All right, so they're showing that this enhanced system's picking up things that are harder on the signal to find when they compare mouse with xenografts as well as initial cell. But they got an unexpected surprise, which is the GTPase Rho-G. And it's been linked to a monogenic form of immunodeficiency, but loss in here is detrimental to normal immune function. So loss of it is normally detrimental to immune function, but it's a strong benefit in the knockouts for CAR T cells. And each of these genes rapidly expanded and account for 25% of all CAR T cells in the mouse system. So I forgot to mention, when they do this mouse system, they throw a bunch of different cells in the mouse in the presence of cancer and go, what knockout ones are expanding better? And they can find that when the downstream sequencing screen, these lineages with these knockouts from the CRISPR, they're the ones that have a survival advantage in Mr. Myces. So then they show that it enhances in vivo. So they go after Rho and FAS. FAS is kind of control, and this Rho-G is the new experimental one. And they both enhance the function of CAR T cell therapies in the mouse system. And a double knockout's even better. The FAS aligns with previous data, so they're using it as a control. They aren't sure how at this point, right? And so they showed that essentially they're more proliferative, which is what you're looking for, but they look at transcriptional differences, and there's more elevation of S phase markers. So like, great, it's growing faster. But what is it doing? And they really, they can't nail exactly what it does, but they figure out that the knockout enhances proliferation, promotes a central memory phenotype, reduces exhaustion, all these good stuffs. So they do a combinatorial screening for synergistic genes, and they find other stuff related to cell cycle and safe harbor locus, and then other essential genes. And they show, hey, that basically the system works and you can find something new. But then they do base editing for clinical translation. So regular CRISPR, right, is a break and can have issues, but there's a single base editor for CRISPRs. They just change a single nucleotide. They don't cause double-stranded breaks. They're single strand to fix. And they do this for saturation mutagenesis of Rho G to find critical amino acids. And they link it then to the missense mutations in GTP binding pocket. So it's related. So its catalytic function is important, right? Which is important to know. So it's a GTPase, and that is what's important about it. They didn't get quite to exactly what it does to generate proliferation, but they got all the way down through their CRISPR technology to a new gene and what part of the gene's region is important for its function in this case. So that was pretty cool. I don't really appreciate how hard it is to do the screen, but it seems like they did a lot of work to get there technically.
Indeed. Yeah. So I guess one of I think there have been other screens on CAR T cells, but most of them are growing the cells in vitro. And I think the advantage of these studies that they're actually putting in our mice, right?
Mm-hmm. Yeah. So
Yeah.
so the advantage is they look at in the human cells systems, but then they can put a screen pile with 50,000, not 50,000, but all the different knockouts into the mice that has a xenograft, and then pull the cells out and measure which ones survive more.
Yeah. Yeah. Yeah.
And they really have very high CRISPR efficiency, which is like, I think the technology part, right? They're able to do this with high enough fidelity that they're getting full genome coverage.
Yeah.
And then finding subtle things by doing it in this natural experiment where the mouse is causing selection over time for expansion.
Yeah.
And so you can see things that take a little more cooking, a little baking in the oven, so to speak, to arise.
Well, it's interesting because right next to that article you just read, there was another publication from from corresponding authors Robert Manguso and Marcella Maus at Harvard Medical School and Broad Institute. First author Nelson Knudsen. And in this, I guess it's not a coincidence they are back to back at Nature, because in this case they are doing something very similar, but I think the results are a bit different, and also the approach is a bit different. So I think it's a nice opportunity to compare how two different groups are kind of trying to answer the same question. So this is again in vivo CRISPR screens identify modifiers of CAR T cell function in myeloma, also published in September 24th. And in this case, again, they're looking for genes that can be knocked out to improve the functionality of CAR T cells, and they look at that in different ways. So one of the ways, the first ways in which they look into this is if they infuse these CAR T cells in mice, again, immunodeficient mice, how, and they do a pull library screen, and then they try to pick up which are the cells that are there after some time. And for that, I think they're doing a bit different, because in this case, they develop what they have the platform is a human. These are human BCMA. I think yours were CD19 cars, right? So this is BCMA cars targeting a mouse xenograft model of human myeloma. And in this case, they do a little bit different. They have a CRISPR, a single guide RNA library. They call it the Mario library. And in this case, they're only targeting on 135 genes. And they do this because then this kind of accounts for the estimated engraftment of how many cells they actually think they will engraft per mouse, how many cells they want, what is the coverage that they want for each size guide RNAs per cells. They want at least 100 cells for each single guide RNA construct per individual human-bearing mouse, so that they think this is important to keep the numbers kind of statistically relevant. So what they do is they're going to sacrifice the complexity of their library. And the way they design this is that they have a dual single guide RNA cassette. And they say they have the single guide RNA of the library cloned in the first position, downstream of a U6 human promoter. And then they have a single guide RNA targeting the TCR of the cell downstream of of a different promoter. And the idea is that they can follow the cells that have integrated this cassette successfully through the deletion of the TCR, and they have basically depletion of CD3 positive cells because CD3 depends on the TCR expression to be on the surface. So this is a way that they use in order to kind of select for cells that definitely have a guide targeting something. And then I think they introduced the CAR, I think lentivirally. And they inject this in the mice. And then they analyze after certain days. They have different standpoints to see which are the genes that are represented differently in different times. And long story short, I want to say they find that, again, they find some expected genes, which indicates that this is a robust procedure, a robust model. But then they basically zero down on one gene, which is CDKN1B, and they show that this is one of the top targets that are found in the cells in kind of the long-term expansion after, I think it's 21 days, what they call late in vivo. And they show that this gene promotes CAR T cell persistence. I think that they mention this in different ways. They also look at in the tumor model. They also do some analysis in vitro, and basically show that these CAR T cells display superior anti-tumor activity, increased survival compared with kind of T cells that don't have this knockout. And this is not the case because they also find other genes that are, for example, beneficial for the in vitro growth, but then they don't really help in the long term for the actual tumor control. And they don't see, so for example, they find another gene, RASA2, and they increase the expansion in vitro, but then they don't really get enriched in the in vivo situation, and they don't have any improved anti-tumor efficacy. And so it's important. I think that's why they show that it's important to be able to tell in vitro expansion versus kind of in vivo survival and functionality. So I think in the end, what they suggest is that CDKN1B seems to be a good target to knock out in CAR T cells, and this will improve their effectiveness. To be honest, I don't find it that surprising because CDKN1B, the protein that is encoded by this gene is a kinase, is a protein that's involved in regulating cell cycle progression, because it inhibits cycling CDK complexes. So basically, by inhibiting these complexes, it prevents the movement through the cellular proliferation phases. And so I guess in a way, it's a tumor suppressor. So I'm not so surprised that this helps the CAR T cells proliferate more in vivo. And I'm not sure, how much people will be willing to knock it out, given that it's kind of associated as a tumor suppressor. But I think that this kind of study shows that, a method by which you can look into different genes, understand see if you can find some new novel genes that might be useful for improving the survival of these CAR T cells in patients. And definitely was a lot of work done invested in this project. And I think the results were pretty robust. And I think they really show the importance of doing in vivo studies and how some genes that are attractive in vitro might not be so much in vivo.
All right, Brenda, but you're the CAR T expert. Which method was better?
Yeah, but I'm surprised that they find different things, right? So they're both meant to, in a way, study the engraftment of the CAR T cells in these mouse models.
Expansion in this case.
An expansion. Yeah, but I don't see any of the genes you mentioned in any of the lists in this paper. And I don't know, did you get PTPN2, for example, was a RASA2? I mentioned SOX as one.
Yeah, they had a whole pile of ones that also were there, but they didn't focus on it. And I didn't go to the extended. Like a lot
Yeah.
of the obvious things that were already known, it found, but that was how they validated that it worked.
Yeah. Yeah. It's funny because in this one they have one of the genes is like IL-2 receptor alpha, and they have different methods of expanding the CAR T cells in vitro. They have either IL-2 or IL-15 and IL-7, which are two kind of, I think there's a lot of like question what is the best cytokine to expand T cells, and I think there's like IL-2 side, there's the IL-15 IL-7 side. IL-16 IL-7 has been suggested to improve the kind of the final phenotype of the cells. I don't know. I think that has, I'm not sure that's so clear anymore, that motion is so uh, supported nowadays. But it's funny because they say, well, IL-2 receptor alpha, if you knock it out and you have the T cells on IL-2, then they don't grow very well. It's like, oh yeah, but you can fix it if you put them in IL-7, IL-15. It's like, okay, that makes sense. Feels a bit obvious, but I guess, yeah, it just, it's also good to see expected things.
Yeah, that's interesting.
Uh,
All right.
yeah.
Well, what's also interesting is our chat with Dr. Robin Orozco at the University of Kansas in just a moment. But before we get to that, have you thought about how long it takes you to isolate immune cells for your research, Brenda? How long?
All the time. Why won't they just separate themselves?
Yeah. Well, maybe you could speed things up. EasySep is a column-free technology that allows you to isolate highly purified immune cells from virtually any sample source in as little as 8 minutes. Immunologists around the world choose EasySep to isolate immune cells and accomplish more with their time in the lab. Learn more at www.stemcell.com/choose-EasySep.
For the second part of our show today, we have a very special guest, a friend of the show, Dr. Robin Orozco. She is assistant professor at the University of Kansas, and she's going to be talking to us about the work that at her lab. And I'm very happy that she's joining us. We met at Hawaii at the AI meeting, and we met her team. She brought many of her students with her. It was such a fun time, and I'm so glad that now we get you here to have a more extended conversation about your research interests. So, Robin, welcome to the Immunology Podcast.
Thank you. I'm excited to be here. Like you mentioned, Brenda, yeah, I'm a long time listener, so this is exciting for me.
All right. Well, first question. So your overall view, kind of your work, right, is you look at single nucleotide polymorphisms or allele variants to understand basically human immunology, right? Like, okay, we have this variant. It's a natural experiment, as you would call it, right? It's just very normal human variation that can tell us things. How has this been useful? What are we able to understand from this that is really helping us fundamentally go, oh, well, that's how all this works?
Yeah, so I think to start off, so I don't do human immunology yet, but I get inspired by human mutations we see in humans and model them in mice. So I don't want to start up people thinking I'm using human cells. So I use mouse models to address this. I think one thing that got me interested and started really thinking about this was the fact that in mouse models, we often take, I would say, more a sledgehammer approach if we just knock out genes. And those are really good approaches, and that's taught us a lot about what different proteins are doing to regulate immune function. But often, these mutations that we have in the human population and just in animals in general aren't knocking out the gene, they're just altering function. And so I think by studying this, it helps us understand a little bit more of the nuance that's occurring in immune cell function and immune cell regulation. One example of this that we study in the lab is with a gene called PTPN22. And historically, people have studied this by knocking it out, which again is a great way to study what is it doing. But humans have a variant in this gene, about 5 to 15% of humans do, depending on which population you're looking at, that doesn't cause the protein to go away. It's still there, just has altered function. And because our lab has been studying this, we've started to understand, I think, some fine tuning that occurs with T cell receptor signaling that, maybe wasn't appreciated as much before. We're seeing some interesting antigen-specific T cell function with this mutation versus with the knockout, we see less nuance that's occurring. And so I don't have like a big example of like, because we studied this completely changed immunology, which I'm sure those examples are out there. I really do think by studying this, we can get to that point and help us understand what's going on. And I think I often tell my students, even though we're studying the fundamentals and kind of basic immunology here, it does impact how we think about treating disease. So, and again, in PTPN22, if we wanted to target it, we would likely target the catalytic domain. It is a phosphatase. That's what people want to go over. But that doesn't knock out the protein. There's still, it's about an 800 sub-amino acid protein. There's still a lot more amino acids there that are not being affected that still have function. So maybe we need to go after those functions too, depending on what we're doing. We're not going in and cutting out PTPN22 from people. I hope that answered your question.
So maybe I'm just going back into where do you find this protein? So oftentimes these kind of genes that come up in genome-wide association studies. I don't know if it's a focus for your group, for your research, but maybe something that you want to share with us is, how do we find this? What does it mean finding a gene in a GWAS study, and what kind of signal you need to find in order for it to make it worth it looking further into this?
Yeah. So we don't do GWAS studies in our lab. I rely on much better genomicists and population geneticists than myself to carry out those studies. But when I'm thinking about what gene or variant do we want to study next, the kind of, I guess, filter that goes through my brain is this gene or variant causing a change in protein function? Is this gene or variant something that is this gene expressed in immune cells? And do we know, because of those knockout studies that might have been done, that it is actually regulating immune cell function? Because at the end of the day, I'm interested in immune cell function. So if it's not regulating immune cell function, I'm probably not going to study it. And then from there, I start to look at, is there, in those GWAS studies, what did they link this variant with? Was it linked with increased risk of different autoimmune disorders, which the variant we study in PTPN22 is associated with that? Has it been studied in the context of looking at a protective effect against different infectious diseases like malaria or Ebola virus, or pick your favorite flashy disease there? Or maybe it's been studied in the context of cancer. There's different studies that are coming out with GWAS trying to figure out single nucleotide polymorphisms, or SNPs, linking with that and regulating immune cell function. So I start to go through those kind of filters. And then I dive into, okay, is this mutation, has the actual variant been well studied? Or is it mostly the knockout, and has it been studied across different contexts? So again, with PTPN22, which I'm going to come back to a lot because that's the only variant we're studying right now, it has been well studied in how it predisposes people to autoimmunity. But it was not well studied when I started my postdoc and started looking at this variant and how it could potentially impact infectious diseases and cancer. And since then, which was 8 years ago, about, more studies have come out looking at that. And I think because we're looking at it in a new context, we're figuring out nuanced things and more specific things about how it is regulating immune cell function. So because of a lens of studying it through autoimmunity, people have largely ignored its role in innate immune cells. They've mostly focused on T cells and B cells because they're main drivers of that disease. So we haven't looked at it in, let's say, natural killer cells, for example. And so I'm going to put a little bit of a plug. We have a paper in revision right now looking at it in natural killer cells. So, I think because we studied this in a new context, it really led us to starting to understand different aspects of the biology. So that's kind of my filter and kind of an example of where it led us to start understanding this polymorphism in a new context and in a cell that it hadn't previously been explored.
Yeah, so you mentioned it's an autoimmune associated allele, but you found other important uses or functions of the mutation in immunity and protection. So could you go over a little bit about what is this allele do to the protein, and then at the biochem level, and then what are you finding its importance in, why we have this allele? Because, things that just make you worse don't tend to survive evolutionarily. So you're finding some benefit to this, and could you go into that a little bit?
Yeah, so Jason, that's a great point, and that's why I started studying this in virus infection, because autoimmunity is not a great selection pressure for keeping something around. So the question was, well, is it doing something protective? So this mutation, it causes an arginine to a tryptophan amino acid substitution on the more C-terminal side of the protein in a domain we call the P1 domain. And in this C-terminal part of the protein, and actually for about 2/3 of the protein, this protein is intrinsically disordered, which means that it is a wet spaghetti noodle flopping around the cell. So it's a nice structured catalytic domain, and then it's just a cooked spaghetti noodle moving all around, this intrinsically disordered. And this P1 domain is in that intrinsically disordered region. So changing from an arginine to a tryptophan is not thought to drastically change the structure of the protein or the stabilization of the protein, because it's moving around, it's highly flexible. So this P1 domain is really important for PTPN22, or that's the gene. In humans, it's called LYP, L-Y-P, and in mice, we call it PEP, P-E-P. So if you hear me use those terms interchangeably, that's what I'm talking about. But this P1 domain in PTPN22 binds to a variety of other proteins, including CSK, which is expressed in T cells, among other cells, TRAF3, which is expressed in all of our immune cells. And when it binds to these proteins, like CSK actually traffics it up to the plasma membrane of an activated cell, where then PTPN22 can use its phosphatase function to dephosphorylate other proteins like LCK and ZAP70 to mediate TCR signaling pathway. When it binds to things like TRAF3, it's thought to be acting in a different way, where it's more kind of TRAF3 is maybe sequestering PTPN22, or maybe PTPN22 is sequestering TRAF3 from certain signaling pathways, changing its ubiquitination status, and that is independent of its phosphatase function. And then PTPN22 can interact with proteins like GRB2, GRIB2, GRAB2, however you want to say it, to mediate other signaling pathways. It's been shown to mediate cell migration and adhesion through its interactions with VAV and the LFA1 complex. So I'm saying all this to get to your point. Biochemically, what is it doing? It's doing a lot. And so that's why when we think about knocking out PTPN22 versus just studying this one variant, can have drastically different effects. There was also a paper that came out a year or two ago that showed PTPN22 promotes PI3 kinase signaling, and that happens independent of the P1 domain. So having this mutation, that autoimmunity risk mutation, we don't think should affect that. So we're seeing these context-specific things. So how does that all play into disease? There's a lot of these biochemical changes that are happening. If we even just focus in on the T cell, we're impacting migration and adhesion, we're impacting TCR signaling. Papers have come out that say it could regulate TGF-beta signaling as well. Taking all those together can certainly progress autoimmunity. But I've shown in my postdoc, and now my group is showing now, that those same changes with some nuance there protects against a chronic virus infection, LCMV clone 13. It also mice that have this mutation have reduced tumor burdens of certain tumors. That's been linked back to the immune response. And that paper I mentioned earlier is coming out. We've actually looked at it's causing protection against coronavirus as well. So there's this balance. Maybe you're more likely to get autoimmunity, but you're potentially going to protect yourself from certain cancers or certain virus infections as well. And the mechanisms by which that occur, some overlap, but some can be distinct as well.
Yeah, it's clear that you found, well, there's many of proteins like this that seem to be in the crossroads of so many different pathways. Must be pretty challenging studying, like trying to separate the different potential influences.
Yes.
Well, I guess a way of doing that is by testing different mouse models, because in this case, you have a nice mouse model that has this mutation that allows you to, study in the context of different diseases. But if you were to try to find, how would you go about looking for more of this, for new variants, or is it a situation in which you cannot use a mouse, or you can, maybe you don't do so much human studies, but it looks like a lot of work, and there must be so many different alleles or so different variants that available to study. Are there other methods that also make sense to use to kind of, oh, you have to go to the mouse? Is that the only way to understand this the complexity of signaling?
Yeah, I think it depends on when you get down to the specific question you're asking, because the fact is humans have tons of genetic variation.
Yeah.
And all that comes together. And so just because I like to study variants in isolation so I can really understand what that is doing. And I'm early in my career, I'm an assistant professor, so I would love, kind of a long-term goals. If we can figure out how one mutation is really working in a specific context, then if we look into the population genomics type studies and see what other mutations this mutation is commonly linked with, which there are ways to do that, then in our mouse model, we can layer on that second mutation and third mutation and start seeing and really teasing out, are they additive in nature for certain signaling pathways or progressing certain diseases? Are they fighting against each other? And then we can start in a almost reductionist way, starting to get to these polygenic type questions. The other approach is that you just make sure you have tons and tons of different human cells or outright mice or something like that. And you just, you screen for individuals who all have what I'm going to call the wild type allele, so this non-autoimmune risk. And then you use CRISPR-Cas9 to induce it, and you just take into consideration all the genetic variation that exists. With that, you're going to need a really high number and really high-powered study to see if what you're seeing is truly PTPN22 mutations or whatever variant. And that becomes a very complicated, messy analysis that at this point, I'm not touching with a 10-foot pole. It's not quite what I do. I'm going to stick to my more reductionist approach. But just because it's reductionist and we're studying it in isolation, I don't think that lessens its impact in trying to understand what's going on.
Yeah, but you have to reduce it. Otherwise, it's impossible. It can
it's
get very overwhelming very quickly. I'm just thinking about it. It's like, man, you have to kind of decide. That's always in so many experiments. Like, I cannot look at everything at the same time. I have to.
I figure it's complicated enough in a mouse
Yeah,
and
exactly.
like a comparable genetic background. Like, we're not switching microbiomes. We're not adding additional genetic variation. We're keeping everything as constant as we possibly can to just look at this one mutation. And it's still different, right? We do use both male and female mice to, see if there's biological sex effects or anything like that. But that there's enough immune cells to keep us busy for quite a long time, at least for this foreseeable future.
So you mentioned therapy earlier. Is this protein target being looked at for therapy, and does the allele have any implications for that?
Yes and yes. So we don't have anything for humans yet in terms of like a drug or something like that. But there is one small molecule inhibitor that seems to be quite specific that we can use in vivo for mice that was developed out of a group from Purdue. And people have been using that to see if we inhibit PTPN22 in different cancer models, if that's a viable therapeutic. And so Elizabeth Jaffe and Wan Jin Ho's group out of Johns Hopkins a few years ago showed that if you give mice this PTPN22 inhibitor, they are able to reduce their tumor burden, and that's associated with a changed immune landscape in the tumor microenvironment, largely the T cells and macrophages. So this inhibitor targets that phosphatase domain. So people are looking at that. People have also showed that individuals, so at the human level, that have this mutation do have decreased risk of numerous cancer types, including melanoma, colorectal cancer, bladder cancer, some CNS cancers as well. So how or why those individuals do have lower risk of developing these tumors, we don't know. That's something we're working on. We're trying to figure that out, again, using our mouse models, as well as some maybe once we figure out in the mouse models what's going on, translating that to the human population. So I think there's promise here to bring targeting PTPN22 into the clinic someday. But there's a lot of work we have to do before we get to that point, including making a very specific inhibitor for human PTPN22. Also, the inhibitor as it is right now has a fairly short half-life. So there's a lot of work that still needs to be done that I know is being done, and it's but not in my lab. We just work with some of these labs to get the inhibitor and try it in our own systems to see outside of cancer, could it also be beneficial in other situations? But then the question would then become, if people have this mutation, do we also give them an inhibitor to a very different part of the protein, and would that still be effective or not? How would it pair with the immunotherapies that we have currently approved or in development for cancers as well? We don't know the answers to those questions quite yet.
It can get very tricky. There's so many, getting an inhibitor, for example, sounds kind of trivial, but it's actually really, really hard.
It's very
So
hard. Yeah. It's especially this is a phosphatase, and if people are not familiar with phosphatases, those catalytic domains of the different protein tyrosine phosphatases are highly conserved across the different phosphatases, and so you want to make sure your inhibitor is specific to your phosphatase of choice, not hitting all of them. In this case, maybe you do want a broad spectrum phosphatase inhibitor.
Yeah.
And so I make my plug here. I study PTPN22. A high related phosphatase is PTPN2. They aren't the same, but they are implicated in very similar pathways. But they've been shown to have non-redundant roles as well. And their catalytic domain is quite similar because they're both PTPNs.
Right.
And so that's one where, if we're targeting PTPN22, you don't also, maybe you don't also want to hit PTPN2 and vice versa. So it becomes complicated, and I can appreciate the chemistry, but I'm certainly not doing that chemistry. But I'm glad people are.
Yeah, indeed. Yeah, no, that must be. I would also.
We're saying drug development is difficult.
Yeah. We've said it.
Should I go on the record and say that? Yeah, I think it is. I have those massive screens that people are doing. I'm part of a chemical biology of infectious disease group at University of Kansas, and so a lot of the peers that I talk with are in thinking about this drug development, and I have seen more chemical structures in my few years here at Kansas than I think I saw in all my education together. And just talking to these chemists about just changing, certain side chains, which changes the permeability of these drugs into the cell and their half-lives and, the pKa's and all this work that goes into it. And even just synthesizing these drugs in different ways. It is quite an effort. So yeah, I'll say it's hard. I think it's hard. I think it's, and that's something that we always appreciate as biologists, all the immense amount of work that has to go into that. And then as, an immunologist and biologist, I just get to reap the benefits of that hard work and then just start, like, oh, does this help protect during virus infection? Does this reduce my cancer? And I'm not diminishing the hard work that me and other immunologists do at that level. It takes, a team, but yeah, I think it's easy just to, grab something. This, you have this little powder that, your chemistry friend across the street gave you to try in your cancer model. And you're like, oh, you just made that, right? No, not quite.
Very easy to make.
Straightforward, yeah.
I wanted to change gears a little bit because when we met at Hawaii, one of the things that struck me about you and is that you have a really nice work group. You brought your students with you, and I just want to have a little bit of conversation about also the teaching and your work as a teacher at Kansas University, and how is that a part of your kind of your job there, and how do you like empowering your trainees? And because I think I have the impression that's something that really matters to you, and I would like to give you a chance to talk to us about that.
Yeah, so teaching and mentoring matter a lot to me. Part of why, so my job for, structure is what's called a 40-40-20 split. So I have 40% teaching effort, 40% research effort, 20% service effort at my university. So I like that because I like teaching undergrads and I also like doing research. So mentoring, I'm going to start with, is really important to me because I've had, I think, really wonderful mentors throughout my whole career. I've been really fortunate that I have not what I would consider a toxic mentor. I've not been in a truly bad lab situation where I felt unsafe or anything like that. So I'm really grateful for that because I know people have gone through those situations. So I started doing research as an undergraduate in a small university, and it was there that I saw the importance that undergraduate research had in my life and other people's kind of careers. So in that lab, which was Dr. Devavani Chatterjee's lab. So if anyone's listening to this and is, big in more undergraduate immunology education, they're familiar with Devavani. She's a great undergraduate immunology educator. And she had this setup where undergraduates were doing research. We had research questions, we were developing methodologies and answering those questions. And I follow that model now as a PI of a lab. So my undergraduates are doing research questions. They're not just in doing lab chores. Everyone, including myself, participates in lab chores. Although I probably participate in less lab chores than everyone else. But so they're doing that. And I think it's important, and I really enjoy mentoring and seeing students work through that process. I often remind my graduate students and everyone in my lab that we're humans first and scientists second. And so embracing that humanity, I think, is where I really like that mentorship. So kind of a pay it forward. I've had great mentors. I want to try to be a supportive mentor as well. And help people work through these problems. And then we can use that positive energy to do really great science. And there's satisfaction in just seeing people succeed. It's a really great job. For my teaching, I teach undergraduate immunology at KU. My class is mostly juniors and seniors, a lot of pre-med, some pre-vet or other health, and just people who may want to go to graduate school. My class is 136 students, so it's quite large. It's not the largest class that's ever been taught, but it's certainly not the smallest. And I teach what's called a flipped format class. So the students watch a lecture ahead of time, then they come to class, we review that material in about 10 minutes, and then we have in-class activities to cement that knowledge. And I like teaching that way because it allows me to really interact with the undergraduates and teach them and have more of a rapport with them and have conversations. And it's really fun. And so I see this and I see, students grasping immunology concepts. So again, today was T cell development. So when they finally, grasped that positive selection and central tolerance is really important for T cells and to preventing diseases. And they're like, I get it. And I'm like, okay, good. And, on Wednesday, we're going to go through B cell development, which always throws them for a loop. But I don't know, I really enjoy it. It also helps me get out of the weeds of my own research quite a bit. When I'm sitting here and thinking about all the ways PTPN22 could impact every single, signaling pathway known to man in every single immune cell, it helps me step back and kind of be like, why does this matter?
Yeah. Perspective.
It's good perspective. Yeah.
That's wonderful.
To wrap this up then, and to keep on the sense of mentoring, to end with what we call our fun question, but I think this is really germane. What is the single best piece of advice you've gotten, either professionally or personally, that is kind of your North Star?
So this is actually advice that came from my postdoc mentor, Linda Sherman at Scripps Research, who's now retired. I'm going to preface this advice with a little bit of a story so people are not ever. At no point in my career have I been tempted to, nor have I been around people who are tempted to truly form like academic misconduct. So this advice is not about academic misconduct type stuff or like falsifying data or anything like that. But Linda and I had gotten in a conversation of where, maybe I'm going to call it gray area, where you see some people being really, really big sticklers for certain things about, how you spend grant money and exactly what you said, versus some people might say like there's some leeway there, maybe how much you spend to sell your story to fit within a specific granting agency, how you treat others in your lab and other research labs and stuff like that. Linda and I were talking about just kind of the morality of science in general. And, I think there are very clear lines that we are not crossing. But I was talking to her about this and she said, Robin, at the end of the day, whatever you decide, it's your life and you have to live with those decisions. And it really struck me because to that point, I had not thought about how decisions I make in my job and my professional career, I take them home with me at the end of the day. So wherever my moral compass is, which I like to think I have a pretty high standard moral compass, that's how I run my research lab. That's how I run my life. And that's different for everyone. And so when students are upset because, oh, well, our lab makes sure we do ABC, certain things by the book. You can think about this in animal care. I have really high standards for animal care and how we do animal research in my lab. And I will admit that they are higher, they are beyond what other people may do, have standards for. Like, well, their labs slack off, in quotes, on this thing. I said, yeah, but that's their morals. Like our standard is this, and because at the end of the day, I have to live with this decision and stuff like that. So I kind of always filter through my own morality that I don't think I hear a lot of people talking about in science, where there's these gray areas. So I guess, yeah, it's you have to follow your own moral compass, which is translated to a lot of things, and is in my work, and also led to a lot of satisfaction in my work, because if I feel confident in my decision that was the right decision based on my own moral compass, that I can feel good moving forward.
Yeah. Stand behind your decisions and make good decisions and then stand behind them.
Yeah, make good decisions. Yeah. Like, this is not like, oh, I feel like, again, that's why I wanted to preface with like, there are clear lines that we're not crossing, and I don't think people should cross, whether you feel fine about it or not.
Yeah.
But yeah, just because someone else goes into an area you're not comfortable with for your own moral reasons doesn't mean you have to follow them.
That's good advice. I like it.
All right. Well, with that, thank you very much for coming on and full circle here.
Yeah.
Yes, thank you so much for having me, letting me nerd out about PTPN22 and genetic variation a little bit and talk about mentorship. I appreciate it.
It was a wonderful conversation. So thank you so much, Romy Orozco from the University of Kansas, and today at the show. And so, well, it was a great conversation. Thank you for joining us.
Yes.
Well, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast, on Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.